the last lecture, I'm just covering some of the things which have not been so far discussed or covered in the last 9 lectures. Ladies and gentlemen, today we are talking about something of the leftover of the renal physiology. Now, that leftover is quite important for examination purpose, and at the same time, quite important for your clinical years. And at the same time, some of the things are very important in your household. Your parents or your seniors might ask you, what is this kidney transplantation? What is this artificial kidney? Right? These are the important part which we are going to discuss today. We shall start with the renal clearance. Renal clearance is something. Now, renal clearance means the volume of the plasma. Ladies and gentlemen, just try to understand me, right? Be careful. We are talking about, when we talk about the renal clearance, that means the volume of the plasma that has been made free from a substance in 1 minute time. Some substance is present in plasma, and that substance is cleared in 1 minute, and cleared from which volume of the plasma? Has the plasma made it totally free? It is not the case like that. But we calculate it, and this is defined as the volume of plasma that has been cleared of a substance in 1 minute. Now, ladies and gentlemen, I'll give you the example. It's somewhat like this. Every minute, nearly 700 ml of the plasma flows through the kidney, right? Out of that, say 125 ml of the glomerular filtration is produced. We are not concerning much about that. Now, we are considering 700 ml of the plasma flows through this. Suppose the urea concentration in plasma is, say 30 milligram per 100 ml of plasma, right? That is, every minute, say 700 ml of the plasma flows, and plasma contains urea, which is concentration is 30 milligram per 100 ml. Now, every minute, suppose in the urine, only 15 milligram of the urea is obtained, cleared off. That is, only 15 milligram of the urea is excreted every minute in the urine. It is coming from plasma, and the plasma contains 30 milligram per 100 ml. That means 50 ml of the plasma has been completely cleared off. Let us divide hypothetically plasma in 2 groups. That is, a part of the plasma is totally free. There is no urea in that one. And there is another part of the plasma, that is the 50 ml of the plasma, which contains 30 milligram. That is, out of 100 milligram, 15 milligram has been cleared out. That is, the 50 ml of the plasma has been completely cleared out, and remaining 50 milligram and 50 ml is not cleared out. So this is what we call it as this is the volume of the plasma that has been cleared. Of urea in 1 minute time is known as urea clearance. And this is expressed in terms of milliliter per minute. That is, you can calculate it, U into V upon P. That is the milligram of the substance excreted per minute. That is the concentration of the urea in urine and volume of urine. That makes the milligram of the substance excreted per minute divided by milligram of the substance per mil in the plasma. So this is how we calculate it. Clearance tests are done to assess the functions of the renal glomeruli and renal tubules. That is the primary purpose of these clearance tests. Coming to the next one, that is the renal clearance test. Clearance test means the volume of the plasma that has been made free from the substance. That is the glomerular function test is to assess the glomerular function we carry out. What? We carry out a test known as inulin clearance test. Ladies and gentlemen, just try to remember the spelling of inulin. It is like insulin. It is not insulin. It is inulin. It is a polymer of carbohydrates, and that polymer is not absorbed and not metabolized in our body. When it is injected in the blood, it is completely filtered out in the glomerular filtrate, right? And this is the test for glomerular filtration rate. That is, it is not normally done nowadays. The reason being is that it requires a constant infusion of the inulin solution through the veins and maintain a certain stable level of the blood concentration, right? And therefore, it is not done, but experimentally it is done in the experimental animals and in human being as well. So this is the test for filtration, glomerular filtration, just to measure the filtration rate, GFR, and it is 125 ml per minute. Right? The next one is the creatinine clearance. Creatinine is a product of our body. That is as a result, muscle contains creatine, and that is as a result of muscular activity, the creatinine is produced, and the level is always maintained in the blood. Right? Now, try to remember normal creatinine level in our blood. It's quite an important one to remember. It is 0.8 milligram to 1.5 milligram. That is normal level. This is the plasma level of the creatinine. Maximum is 1.5 milligram. If it is more than that, then that shows the kidney functions are disturbed. Now, creatinine is a substance. Creatinine is a substance which is secreted by the renal tubular cells, and then it is added to the glomerular fluid. I mean, say tubular fluid. So this the normal. This shows if the creatinine clearance test, if the level is up to 110 milliliter, that means kidney is functioning normally, right? And then we come to PAS. This is a new substance, para-aminobutyric acid. This is primarily responsible for the measurement of renal blood flow. Indeed, we are measuring renal plasma flow, and when we calculate on the basis of hematocrit, the total blood flow. Do you remember that is the 1/4 of the cardiac output passing through the kidney? That means cardiac output is nearly 5 liters. That means nearly 1200 ml of the blood flows through the kidney every minute. That contains nearly 650 to 700 ml of the plasma. This substance is injected. PAS is injected. No, PAS has got a peculiarity that it is completely secreted out of the tubule. So in the arterial blood, it contains, the level is very high. In the renal vein, in the renal artery, the level is very high. In the renal vein, it is very, very low. Almost all have been excreted out by the tubules. The normal value of this one is that is 600 to 650 ml per minute. Right? This is quite an important one, just to measure the renal blood flow, RPL, renal blood flow. This is measuring renal plasma flow, and we can calculate it by hematocrit. That is, normally in the blood, 55% is the plasma and 45% is the cells. To assess the tubular function, urea excretion is again, this is not 8 millimeter, this is the 80 millimeter. This is the 70 to 80 millimeter normal urea clearance. This is a urea is a very strange type of the substance in the body. It is filtered in a huge amount by the global filtrate, and surprisingly, in the proximal tubule, as we have seen, urea is absorbed, reabsorbed here, but it is excreted out in the collecting ducts. In the collecting ducts, by osmosis, a huge amount of the urea is reabsorbed. That is partly responsible to maintain the I mean say medullary concentration gradient. So that is also equally important for the assessment of the tubular functions. pH is again we have taken clear that this is a high abstraction, huge amount of the substance is taken out of the blood and excreted in the urine. That is primarily used for the purpose of measuring renal plasma flow or blood flow. Ladies and gentlemen, this is quite an important one. Concentration and dilution test. Now, if you test urine, you will be given quite a good idea in the biochemistry. That is the urine. I mean, say here concentration and dilution test is that the urine is tested for the specific gravity. Normally, specific gravity we take it as in terms of water. Distilled water specific gravity is taken at 1,000. So urine is compared with the specific gravity of water, and in that one it turns out to be 1,010 to 1,015. That is a normal urine concentration. If you have not taken water for quite a long time, that is in this case, we carry out a concentration test. The individual is not given a large quantity of water, and it is made partly dehydrated. That results in the increase in the blood osmolarity. That leads to the release of, when the osmolarity increases, there is an increased secretion of ADS. ADS will cause maximum reabsorption of water. So urine becomes concentrated. That is, reabsorption of the water takes place in the tubule. So this is the tubular test. That is the in case of concentration test. If the specific gravity of the urine is say nearly 1,025, that is taken as a normal one. That means the concentrating ability of the tubules are perfectly normal. In case of dilution test, the person is given a huge quantity of water to drink, right? And then the diluted urine is produced. Reason being is when the large quantity of water, how over dehydration happens, that is the osmolarity of the blood, osmolarity of the body fluid decreases. That prevents the secretion of ADS. So ADS is not secreted now. So whatever the fluid comes in the collecting ducts will be passed out as a diluted urine. And diluted urine the dilution can reach to 1,005. That is, the kidney has got an ability to pass the diluted urine in the event of the overhydration of the body. And next comes the, ladies and gentlemen, this is NPN, non-protein nitrogen. That is the, normally we pass 1,500 ml of urine in 24 hours. That is in a normal condition. I'm talking about a person who is living in the ambient temperature of 22, 23 degrees centigrade. And say passing 1,500 ml of the urine, 600 to 700 ml in the perspiration, and 300 ml in the respiration. That is, it is this 1,500 ml is enough to excrete all the metabolic waste. So what are the metabolic waste? That is what we call as a NPN, non-protein nitrogen. Non-protein nitrogen product are urea, uric acid, excreted And these are required, minimum requirement is 500 mil. So, ladies and gentlemen, that is, I was talking to you, that is needed in the, in the clinical side. And you say, your prof in the medicine or surgery, they might ask you, what is the minimum amount of urine to be passed by the person to clean all the metabolic waste? Your answer should be minimum 500 mil. If the less than 500 mil, the metabolic wastes are not excreted out. A person, another one is equally amount. Can a person live with one kidney? Your answer should be yes. Person can live well with one kidney. Another kidney maybe removed or maybe donated to somebody. Okay. Even a person can survive with a half a kidney. When the kidney, say 3/4 of the kidney, one full and another half, it is removed and only one half is kept in the body, person can survive. Metabolic waste can be excreted out, but he or she will not be able to go for a stressful conditions, not in the exercise or like that, but it can survive. Another thing is, which you have to remember, nephrons. Nephron means glomeruli and tubules. Can they regenerate? Do they have a power of regeneration? They do. They can regenerate in a short-term injury or mild injury. Injury must be short-term or it must be a mild one. These nephrons can regenerate. Not in the tubular necrosis as it happens in the heavy metal poisoning, right? In the heavy metal poisoning, there's a tubular necrosis, and that cannot regenerate. Ladies and gentlemen, coming to the another important point, what is the uremia? Uremia means accumulation of the uric acid, creatinine, and potassium ions at the toxic level. That is, they are present normally in the plasma, but when they reach to the toxic level, we call it as a state of uremia. That is, urea level, which is the maximum 45, can go to 100. Uric acid level, which is the maximum, say 6.5 or something like that, can go very high. Same thing is the creatinine, which we take it as a normal high level, and potassium. Ladies and gentlemen, try to remember, I'm talking about the extracellular potassium. Normal extracellular potassium is 4.5 to 5.5 milligram, milliequivalent or milligram, right? If it goes above that one, it is quite a toxic one, toxic to the cardiac muscles, toxic to the cardiac rhythms, and therefore this is known as uremia. Ladies and gentlemen, coming to the another important point, which we are going to discuss today in a little greater details, that is the renal failure. Renal failure, it is a condition of progressively decreased urine output. That is, progressively decreased urine making. Passing damage due to damage to the glomeruli or renal tubule, resulting in the loss of renal functions. So this is the renal failure. Renal failure means progressively decrease in the quantity of urine passed, and therefore nitrogenous products and waste products they accumulate in the blood and create large number of it, manifest in the different form. Coming to the renal failure, there are two types, acute and chronic. Coming to the acute renal failure, ladies and gentlemen, acute renal failure is a condition of a sudden shutdown of all the functions of the kidney, accompanied by acute renal failure. Try to remember quite an important one. A condition of a sudden shutdown of all the functions. Kidney stops functioning. And creatinine level becomes, rises more than 25%. We call it as a state of acute renal failure. It is characterized by oliguria. Oliguria means decreased quantity of urine production. Decreased quantity of urine production. Decreased, decreased quantity of urine production, that is less than 250 ml of urine is produced, that is known as oliguria. It may be anuria. There may be no production of urine at all. I mean, the patient is in the hospital and being treated for urea or anuria or something like that, and if he develops a state of anuria, that is quite a dangerous state, right? So these are the two terms. Try to remember. In many cases, now try to remember, acute renal failure may resolve, may become perfectly all right in days or weeks, right? If you treat it in a nice way, in a better way. But it may develop in a state of chronic renal failure. It may go in a chronic renal failure. And chronic renal failure is quite dangerous state. That is coming to the, what are the common causes of renal failure? Try to remember this one, quite an important one. Because, I mean, say, it may be asked in physiology practical or in medicine practical or in surgery or anywhere in the clinical science. The common causes of the renal failure turns out to be injury, blows to the lower back. I may say many a times with the jockeying one, we just give a good blow to the backside of the body, and person may develop a acute renal failure. Hypotension is severe and prolonged. Hypotension, fall in the blood pressure, less than 80 millimeter of the systolic. 80 millimeter of the systolic. That state can cause acute necrosis of the renal tubules. That is, a decreased blood supply is responsible for that one, and that can lead to I mean, say, hypotension, and hypotension can cause renal failure. Heavy metal poisoning. You have to be careful about this one. In case of heavy metal poisoning, usually the poisoning is of the lead poisoning, mercury poisoning, and arsenic poisoning. How do we get it out of that? Ladies and gentlemen, in the rural areas in India and as well as in Nepal and mainly in these two countries, even in Pakistan and Bangladesh, there is some sort of what we call as a salt of these heavy metals. They are known as basam, right? The salt, the powdered form of this one, it is given, and that can cause a severe damage to the kidney tubules. That leads to heavy metal poisoning and renal failure. Blood transfusion, mismatched transfusion. That is, the A group of the blood has been given to B group, and this can cause hemolysis and blocking of the renal tubules. I mean, this is very important one here. That is, as long as the hemoglobin is present inside the red cell, that is a fine. It is very good. Once it is outside the red cell, that happens as a result of hemolysis. It can cause clogging of. It can cause blocking of the renal tubule. Can lead to renal failure. Hypertension, quite an important disease of the modern, I mean, say, modern civilization, right? Hypertension can cause two important things in the kidney, that it can cause arterial sclerosis, and therefore there is a decreased blood supply, and that leads to decrease in the GFR and decrease in the renal function. Another important thing, equally important thing, the diabetes mellitus. Diabetes mellitus is a disease which is the mother of all the diseases, right? In the diabetes, there is there is an atherosclerosis, not the arteriosclerosis. There is an atheroma leading to decrease in the diameter of the blood vessels, decrease blood supply, and that leads to renal failure. And then another thing, quite an important one, that is autoimmune glomerulonephritis. It may be an acute nephritis of the fulminating type, but autoimmune glomerulonephritis is a very, very important disease that is for the purpose of uh, renal failure. Coming to the next one, ladies and gentlemen, we are coming to physiological basis of treatment of chronic renal failure, CRA, chronic renal failure. Try to remember, chronic renal failure. This is done, that is the physiological basis. This is done on the basis of dialysis. Dialysis refers to the diffusion of the soluble solutes from a solution of higher concentration to the solution of the lower concentration. So dialysis means some sort of diffusion. And diffusion of the electrolytes or water soluble substances from a higher concentration to a low concentration. That is the principle of dialysis. Right? And when the solution is passing through the semi-permeable membrane, they are separated by semi-permeable membrane. That is the solution of the higher concentration, and semi-permeable membrane is placed in between, and solution of the low concentration from a higher concentration goes towards a low concentration. That is the normal diffusion of the soluble, and passing through the semi-permeable membrane. Here, what we are doing it here, that is blood from the patients of the chronic renal failure is dialyzed with the dialyzing fluid. Ladies and gentlemen, you have to remember what is the dialyzing fluid. That is, the patient's blood is kept on one side, there is a semi-permeable membrane, and other side is the dialyzing fluid. So whatever in the higher concentration, water soluble and a higher concentration in the blood will flow towards the dialyzing fluid. And whatever in the dialyzing fluid in a higher concentration will go to the blood. So we are putting a diffusible membrane which is responsible for the high level in the plasma. We find in the chronic renal failure patient's plasma contains a very high level of potassium. Very high level of the potassium means potassium level in the extracellular fluid may be say 7 or 8 micrograms, 8 milliequivalent. That is taken as a high one, because normal is high point 5 is the maximum limit, and once it goes up, this becomes high potassium is a toxic to the heart muscles, to the conducting system. So high level of the potassium, high level of the urea, high level of the creatinine, high level of the uric acid and metabolic acid, they are in a high level in the plasma. We have to remove them, and we have to remove them from. Somebody is asking me what is mean by dialysis. Dialysis refers to the diffusion of the solutes soluble in water from the solution of a higher concentration to a solution of the lower concentration through a semi-permeable membrane. Right? So that is the dialysis. Coming to the next one. Patient may have. Now see, we have talked about patient may have high level of potassium, high level of urea, high level of creatinine, high level of uric acid, high level of metabolic waste, right? But will have low level of what? Low level of sodium, potassium, calcium, chlorides, and bicarbonates and glucose. These are the substances which are at a low level in the patient's blood, patient's plasma. Why glucose is low level? Because glucose is continuously consumed. There is no source of glucose coming back. But person is and bicarbonate, because continuously acids are produced. Acid is responsible to produce acidosis, and therefore that lowers the level of sodium. I'm sorry, it is not the potassium here. Potassium level is already high. Chloride and calcium. Dialyzing fluid composition is similar to plasma, except plasma proteins. Except plasma proteins, they are similar to plasma, except plasma proteins, right? And waste products. Dialyzing fluid contains everything about except plasma proteins and waste products. Right? Dialyzing fluid contains a large quantity of glucose, calcium, and bicarbonates. So dialysis is done. This is a kind of diffusion. Dialysis can be of two types: hemodialysis and peritoneal dialysis. What is a hemodialysis? What is a peritoneal dialysis? Hemodialysis is done only in the hospital. That is, a hemodialysis means blood is made free from certain substances and added certain substances to the blood. So hemodialysis is done only in the hospital, not outside, not in the house or ambulatory or in the office. Hemodialysis is a machine, and that machine is known as artificial kidney. So artificial kidney is a machine in which the semi-permeable membrane is there, blood flows. बच्चों में तो ये 30 minutes का था, गड़बड़ हो गया, बीच में बंद हो गया. I'm starting again. So we were talking about the artificial kidney. That is, the artificial kidney is the hemodialysis machine which we have in our hospital also, LMC. Hemodialysis is done here. And this is known as the artificial kidney, right? In this procedure, just try to remember it here. This is a quite important one, because you have to explain it in your clinical side. You have to explain it in your homes, to your friends, to your other members of the society sometimes. So in this procedure, the radial artery blood of the patient is passed through the hemodialysis machine. That is, hemodialysis machine is a coiled cellophane tube. That is the semi-permeable membrane tube. That tube is immersed in the dialysis fluid. That is. That is really what happens. That is, suppose a cellophane tube, cellophane tube, our fingers are cellophane tubes and we immerse it in water. So water is a dialyzing fluid, and the cellophane tubes are present in our fingers, and the skin is the semi-permeable membrane. Right? So this is how the radial artery blood is taken, is passed through the hemodialysis machine and through a cellophane tube. Machine is a cellophane tube immersed in the dialyzing fluid. Now this is passed through the tubes, and therefore the coagulation is quite an imminent one. Heparin is added as an anticoagulant. Ladies and gentlemen, this procedure is done only in the hospital. It takes 3 to 5 hours on that day when it is done, and at the same time it is to be done 2 to 3 times a week. That is what we call as a radial artery. So what is happening here in hemodialysis? In the hemodialysis, all water-soluble substances from plasma except protein diffuse through the cellophane machine into the dialyzing fluid. That is, all the water-soluble excess substances present, they diffuse from a higher concentration to a lower concentration. If urea is in a higher concentration, it will diffuse. If it is not higher concentration, it will not diffuse. So diffusion takes place only on the basis of the concentration gradient. Similarly, the dialyzing fluid, try to remember, dialyzing fluid contains glucose, sodium, calcium, and bicarbonates and drug are infused in the blood. From the dialyzing fluid, That is in the dialyzing fluid. What we add? We add glucose, we add sodium, we add calcium, we add bicarbonates, and any other drug which the patient is needing. We add in that one that will also diffuse to the patient's plasma. That is goes to the patient's blood. Now this procedure, that is the dialyzing through the machine, the artificial kidney, what happens as a result of? So whatever in the higher concentration in the plasma will diffuse to the dialyzing fluid. And whatever in the higher concentration in the dialyzing fluid will go to the plasma, patient's plasma. Now this procedure makes plasma free from toxic waste. Number one, toxic waste. Number two, hyperkalemia. And number three, excess of water. Excess of water is always there in the uremic patient, and therefore it is your advice. You will advise them that they will take a minimum quantity of water. But you will count the drops of water that you are taking, because the water, excess water, may be harmful in the body, and that is to be removed in the dialyzing fluid. Patient is given injection EPO, that is the erythropoietin. Why do we give erythropoietin? Because the kidney is damaged, there is no production of erythropoietin, therefore erythropoietin must be given from outside, otherwise it may lead to a severe state of bone marrow depression and anemia. So to prevent anemia, we have to give them injection of erythropoietin at the time of dialysis. The second one is it has to be given calcitriol, not necessarily in the injection form, but it can be given in the dialyzing fluid. Calcitriol. Vitamin D is finally activated in proximal converted tubules, right? That is cholecalciferol, when passing through the liver, it becomes 25 cholecalciferol, and when it passes through the kidney, in the proximal tubule, it is converted into dicholecalciferol, 125 dihydrocholecalciferol. So activated there, and now since there is no proximal tubule, so vitamin D deficiency can occur. That leads to calcium deficiency, and therefore vitamin D3 activated form must be added to the blood. Ladies and gentlemen, what to do with the dialyzing fluid? Dialyzed blood is returned. Dialyzed blood, that is the purified blood, returned to the patient's body through a vein. That is, this is how the blood is from the radial artery passing through the dialyzing machine and going back to the veins. Ultimately, it goes to the veins. Dialyzed fluid is discarded. The fluid which is dialyzed is discarded. Coming to the next one. This is the principle of the dialysis. This is a membrane, semi-permeable membrane. Here is the blood, and here is the dialyzing fluid. From the blood, the rubbish material, waste material comes over in the dialyzing fluid, and from here, the material which we want to go back to the blood, that contains, that is the glucose, bicarbonates, and other things that goes there. So diffusion takes place through this dialyzing fluid. And then I have tried to get this one. Do you see what is happening here? The blood is passing through this artery, going through a pump, a blood pump, which pumps the blood in the, these are the cellophane, and you say these are the tubes, and immersed in this cellophane, this is the, in between the tube, the fluid is dialyzing fluid. That is the putrid view of the dialyzing fluid inside that one, right? So this is exchanging here, diffusing here, and the whole blood passes through. This is the bubble trap. In case if there is a bubble somewhere in the blood that is trapped here, and blood goes back to the. This is the vein. which we will see, goes back to the patient's body. Into the little detail one here. Now see, this is the artery and this is the vein, right? And we put a stent here. That is, the stent is placed here, and that is responsible. That is, we don't want to puncture the artery again and again, because it has to be done at least 2-3 times a week. So this is how it is passing. This is the blood passing through the cellophane tube, and return back to the vein. This is how the blood is pumped dialysis chamber, where it flows through the selectively permeable membrane surrounded by the dialyzing fluid. That is an important one. Try to remember this one. Coming to the next one. I was talking to you. Ladies and gentlemen, artificial kidney is not a small machine. It is a big machine. You see this is the artificial kidney. This is the artificial kidney. This is the patient. And patient say blood pressure and everything is monitored here, and it is strapped like this. Blood passes through, and somebody is sitting by the side of the patient, keeping the patient calm and short. Right? So this is the story of artificial kidney. Ladies and gentlemen, this is what we were talking about, the hemodialysis. Here is another one that is known as the peritoneal dialysis. Ladies and gentlemen, I don't know whether you have dissected abdomen or not. But in the abdomen, you will find a large number of the organs. And they are covered by a membrane known as peritoneal membrane. There is a peritoneal sac. The peritoneum is act as a semi-permeable membrane. That is, you put something inside that one, semi-permeable membrane, it can diffuse. So peritoneal dialysis can be done even at home. Hemodialysis can be done only in the hospital, but the peritoneal dialysis can be done in the home. That is in this procedure. Ladies and gentlemen, just try to remember, this seems to be quite easy. It seems to be quite cumbersome. Here what we do, patient himself pass a soft catheter in the peritoneal fluid, right? And 2 liters of the dialyzing fluid through that cavity. 2 liters of the dialyzing fluid is passed to the peritoneal cavity through a soft catheter. And this fluid is to be kept there, remains there for nearly 20 minutes. And after that, the fluid is drained back. Now, once the fluid is drained back, it is measured again. Input and output must be maintained in a very strict way. Now, another disadvantage is this is to be done at 6 hourly. That is 4 times a day. 4 times in 24 hours, it is to be done. Whereas in case of hemodialysis, it is to be done after 2 days or 3 days, like that. So this is the little bit disadvantage. Now, it is done 6 hourly, 4 times every day. Strict sterilization and infection control must be observed, because peritoneum is very sensitive to infection, and once the infection is there, peritonitis is there. That is a very, very horrible disorder. It is to be. Now, we are coming to the another important point, the final point. Final point is, some of you are very interested in this one. Kidney transplantation is the final answer to all the problems of chronic renal failure. Chronic renal failure has got several problems, right? I will say high urea level, high creatinine level, anuria and many problems are there. But kidney transplantation is answer, final answer to all the problems. Once the chronic renal failure patient is given kidney transplantation, he or she becomes perfectly all right for quite a long time. Kidney is taken from a cadaver or a close relative. From where do we get kidney? Cadaver means dead body. We can get it from a dead body. We can get it from a sibling or a parents or relatives of the person. Usually kidney is not allowed to be taken from other persons. Usually taken. Now, in this kidney, in this transplantation, histocompatibility of the ABO group and major histocompatibility complex must be matched. That there must be a matching of the blood groups and there must be a matching of the tissue groups. They must be matched. They must be of the same order. Donor and recipient. Donor means whether cadaver or a living person. Recipient means definitely a living person. Patient must be of same ABO and same MSC group. There should not be different group. They say MSC group is different, then kidney is rejected. So kidney is rejected by immunity, autoimmunity. Now before transplantation is done, I mean say this is done, before the transplantation is done, donor kidney is flushed with the preservative fluid. That is donor kidney, that is from the arterial side, it is flushed with the preservative fluid and cleaned out. And perirenal pad of fat is also removed from there. Ladies and gentlemen, coming to the final point, donor kidney which we have taken from the other side, donor kidney is, I mean say, in the kidney transplantation, in the renal transplantation, the diseased kidney is not removed; it is left there in the place. Now, where to place the donor kidney? The donor kidney is placed usually in the iliac fossa, right? In the iliac fossa, and placed retroperitoneal, outside the peritoneum. As the kidneys and the ureter and the bladder, they are retroperitoneal. This kidney is also placed retroperitoneal. The peritoneum is kept in the forward direction and retroperitoneal. Then it comes to what happens there, what to do there. Now try to remember, end to side anastomosis. You know anastomosis? Joining of the two tubes, joining of the two arteries or two veins, like that. Anastomosis. And to side anastomosis. That is means something is ending here and this is the side. This is how the anastomosis is done. So this is what we are doing it here. Renal vein. Suppose this happens to be the renal vein and this is the iliac vein. And to side anastomosis. The renal vein is anastomosed with the iliac vein. Right? So blood from the renal vein can go easily to the iliac vein. Like this. End to side, not the end to end. End to end means two ends are meeting. Here, end to side anastomosis is done. Similarly, end to side anastomosis is done between the renal artery and external iliac artery. That is again this side to side anastomosis. Renal artery is anastomosed with the external iliac artery. So this is done in the pelvis, in the iliac fossa. Now what happens to the ureter? This creates another problem. Right? So ureter, remembering that in the urinary bladder, we have seen outer one is the fibrous layer and then detrusor muscles and then transitional epithelium. Mucosa of the transitional epithelium. So a submucosal tunnel is produced. That is through the submucosa and then through the detrusor muscles, right? A thread is passed, ureter is tied and pulled back. That is what we call as a ureter is brought to the urinary bladder through a submucosal tunnel. That is, the tunnel is formed by the submucosal tunnel is formed by the arterial force, and then ureter is pulled to there, and the end of the ureter, opening of the ureter is stitched with the urethra. So whatsoever the new, transplanted kidney produces urine that can come to the urinary bladder. That is the ureter is brought to the urinary bladder through the submucosal tunnel. That is, ladies and gentlemen, come to this point. This is the kidney. This is the arterial venous anastomosis here, right? This is the iliac artery, and coming to this one. This is the ureter. Ureter is tied here to a thread, and it is pulled through this. This is the forceps. This thread is pulled and brought to the submucosal level, and here it is tied, right? So this is what is happening. This is what is happening. How it is done. So what we are talking about, that is the another important thing here is that is infection is taken care of. Say person should be free from infection for quite a long time. It is to be kept in the isolated, clean room for quite a long time, and at the same time, immune suppressive drugs. To prevent the rejection, immunosuppressive drugs must be given, right? That is, immunosuppressive drugs usually we use as steroids, prednisolone, and cyclosporin. Cyclosporin is immunosuppressive drug, prednisolone is a steroid. This is how the kidney transplantation is done. Ladies and gentlemen, thank you very much. God bless you. Any question that you would like to have? So I will try. This is my last lecture in renal physiology. Somebody will come again here to talk something. Thank you very much.